Good morning, everybody. How are you doing out there? I hope everything is going well. I'm really excited to see that four of us are into the Google Hangout already. So that's excellent. Excited to see everybody there, and hopefully we can get. I think we can get one or two more people in that hangout, then we can actually start to hold our classes as a live discussion, if you like. It's just an option. So we'll put out some feelers there to see if that's something you want to do. I'm very excited to be in the genetics unit. Hopefully, you are as well, because today we are going to really get into it. We're getting right into Gregor Mendel and his whole deal and all that stuff, all that good stuff. Oh, by the way, I did end up getting my skull printed. I have a gorilla skull. It's really quite the shame because one of the things I look forward to every year is the ability to get students to handle these skulls. This is now a life-sized gorilla skull, and like putting your hands on it and realizing how similar and how different it is really gives you some interesting ideas about our own sort of place in the world, and looking at the chimpanzee and the gorilla and thinking about how the traits they have are similar but different. That's also another interesting idea to look at. And thinking about even looking at the way their skulls are attached, and the detail, the size of the eyes relative to the size of the face, and all these sorts of things. And it would have been very exciting to compare this to human skulls, which is the next thing I'm printing, and have a little discussion about our place in this crazy, wild world that we live in. Extra wild right now, but that will have to wait till next year, I suppose, which is unfortunate. Um, and so I will we'll leave that to the side. Maybe I'll post a video about it, and we'll do a little project by distance. Who knows? I forgot to mention my colorimeter that I made for my other class as well. Very exciting as well about that as well. Anyways, let's go on to it. Gregor Mendel. So this is considered at least the European father of genetics. He is a mathematician, sort of scientist of the time, born in 1822. He attended university in Vienna, and he ended up becoming a monk. So he lived as an Austrian monk for many years, and during that time he became very interested in breeding plants. And while breeding plants, he started to track a lot of different details about these plants, particularly these 7 traits about these plants that he was very interested in. So I'm, me, not huge on history thing. I like the context a little bit. It's interesting to think about what were the limitations at the time. Here you have a man whose limitations were that he was a monk, so he didn't have many tools or techniques. He does have training in mathematics, so, and he has some knowledge of math and statistics a little bit. It's 1822 still, so we don't have that much statistics. And so he's sitting there playing around in the garden, really. And as he plays around the garden, he just keeps track of all of the different little details he notices in that garden. So he actually tracked a bunch of different plants, but pea plants are the ones we talk about. Pea plants have some favorable traits that make it a good idea for tracking. One thing is that pea plants reproduce quickly and put out multiple seeds and they grow rapidly. So all of these traits are very favorable when you're trying to uh, study genetics. These are traits that would also be valuable for studying evolution. Thinking about how evolution and genetics are tightly related. We know that evolution takes a long time and multiple generations. So if you wanted to track a species and you wanted to track the different generations and changes in those generations over time, then you would want to choose a species that reproduces very rapidly multiple generations over a person's lifetime, as happened for Mendel. So he noted that for these 7 traits, now not all, there are lots of other traits about plants that if you start to look at them closely, there's lots of things that you could look at. You could look at the shape of the leaves or the size of the leaves, all sorts of other features, or more difficult to look at features like roots and root shape, root size, root design, whatever. Um, Now, he chose these 7 traits because each of these traits seem to only have 2 common possible variations. We probably should put a little tiny, caveat here that we are talking about the common variations. Just in case, because in reality, there probably are, you know, what's the difference between yellow and like an off yellow? There might have been a couple of other variations there, and flowers with stripes and flowers with variations. But these variations were the most common, and he was able to sort of narrow things down and say, "Hey, look, there's this and there's this." And the plants come out this way or that way, or this way or that way. They come out with gray and round seeds, or they come out with white and wrinkled seeds. Oh, they come out with yellow on the inside, or they come out with green on the inside. They come out with white flowers, or they come out with purple flowers. They come out with full pea pods, or they come up with the constricted pea pods. And every single flower, he could look through that flower very carefully once it had been grown, and he could count out all the 7 traits, and he could record that into a little diary. And that's what he did. He essentially recorded for years the a diary full of these traits, and he wrote them all down. He was just very diligent, careful about making sure these traits were all recorded. And the result was that at the end of these many, many years of growing plants, he had a very clear record that he was able to track and look into and try to find a sort of mathematical model that would describe the changes he would see over generations. He did end up making one paper, I believe. Someone can correct me with that at a different point, or you can look it up yourselves. But I believe he wrote Gregor Mendel. I think I have uh, lost and found. There is a book on him here, Genetics Lost and Found. I will post this link into our classroom. Let me just copy that link. Okay, so I'll drop this into our classroom. There's a crash course history lesson on this about how his work was lost to time for a while and then recovered at a later date. And that was sort of an interesting little historical note. So maybe I'll just drop it in there. Here's Mendel Lost and Found. Before I completely forget that did not look like what I want. All right, well, I will get into that later. Okay. keep getting distracted. It's too, without you guys in the room telling me what to do, I'm always distracted here. Okay. So he decides to do these experiments, or his experiments, really just growing some plants. Um, he used them to understand how the traits were inherited by cross-pollinating them. So he starts to be a little bit more careful about his cross-pollination, seeing if he can control which traits are going to happen. So the one thing the most important experiment he probably developed was taking a plant that appeared to be pure breeding, so this is a plant that would always produce yellow seeds, and another plant that would always produce green seeds. And he took these two plants and he crossed them. And what that means is that he took the male or the female gametes from one plant and combined them with the male or female gametes from the other. Usually this was done by taking the male gametes, the pollen, so this is like pollen grains, from the flower over to a green pea plant of the female plant. That's the pistil, I think, of the female plant. And then he would actually go in and with a little pair of scissors cut off the pollen grains on the female plant, or to make the plant female essentially. He cut off the pistils so that the female yellow plant could not self. By doing this little trimming, he was able to make sure that the plants were only mating with the ones that he wanted. I think he did this with a paintbrush, I believe. He just kind of took a paintbrush, painted the little pollen grains off, and then painted those onto the female organ of the other plant. This was called the parent generation. So this is the P generation. It is the first generation. It's the two parents that are being crossed. These two parents produce a generation of plants that, in this case, all had the same color. They were all yellow. So this is what's known as the first felial generation, or the F1 generation. He then took those two plants and crossed them, and he got another generation of plants. So all of these plants got their genetic information from this crossing. Now remember that at this time, Mendel does not know that about inheritance. He does not know that genes are passed from one generation to the next. All he knows is what he can see. And so what he sees is that crossing two yellow plants could produce a set of offspring with a ratio of 3 to 1 on average. Now this is an average ratio. This is on average. So that means that we're talking about over many offspring. This is another thing that this could have only really been discovered through plants. Later it was confirmed with flies, because flies do produce a lot of babies. So they did use the same sort of technique with fruit flies. But if you were just a biologist in 1822, the ability to grow fruit flies and to track them and to control their mating was probably pretty limited, because you probably didn't even have a very good microscope. So for somebody in 1822, this was probably the best experiment that he could have stumbled on. And he actually did try to do a couple of other plants, and the results were a mess. And that's because for other plants, the genetics are not as clean. The inheritance patterns are not very clear. So this was just luck in a way. There are probably, scientists all around the world, or scientists because it was the 1800s, that all over the world breeding all sorts of different plants and not really seeing any patterns. And it was only because Mendel was working with this set of plants that had very strict inheritance rules, just because of the type of genetics they had. So Mendel concluded that there must be two forms of the trait in the plant, and that in some cases a plant could carry the second form even though it didn't appear to have it. So, uh, and so we concluded there's two forms of the plant in every plant of the seed trait in the pea plants, and similarly, conclusions for the other traits. And out of these two forms, the two forms had different behavior. So they had different behavior. One form could mask the presence of the other. Now, the interesting thing is that this conclusion does not just come from like observation. It really, really, really comes from the math. This is a mathematical conclusion. When he saw this conclusion of 3 to 1, he realized over time that there was only one mathematical solution, and that would be a two-variable solution, where combining two variables creates 4 possibilities, and if you concluded that one could mask the other, then that only actually made possibilities and one alternate. So it's really, really a math principle. So alleles, alleles are the name for these two different forms. So the name for forms. And so we have dominant alleles. These ones are the ones that can mask. A form of the trait that will always be expressed. Possibly masking the presence of other form. And then the recessive allele can only be expressed if no other alleles are present. So I might let's do a little bit of conversion here. I always think that I'm writing nicely, and then I look at it and I say, oh, it could be better. Okay. Okay. So this got formalized into a couple of different rules. Some of those rules were some are rules, some are we often call these principles because some people will call them Mendel's laws. because Mendel certainly thought that they might have been laws. They might be things that were always true, no matter what the situation. But in the end, and especially by the end of this course, I am going to try to convince you that there are examples in modern genetics that break all of his rules. So they should be thought of as principles. Even the law of segregation and law of independent assortment are a bit debatable. So, but principles, we can also use these as assumptions. These might be thought of as these are the four assumptions. That's probably the best way to think about them. These are the four assumptions of Mendelian genetics and our Mendelian inheritance. Heritance. So I'm going to talk about Mendelian and non-Mendelian genetics throughout this course, and I want you to be comfortable with that idea. These are the Mendelian assumptions. So it's not too different from, when we were talking about evolution, we had two different models or multiple models of why evolution could occur. We're eventually going to sort of have the same sort of five fingers kind of approach, but with genetics. What are the different ways that genetics can be inherited and that they can have impact on on an organism. So the first one is the principle of genes in pairs. So in Mendelian genetics, we will assume Mendelian genetics assumes that all genes all traits are controlled by a single pair of alleles. So in any individual. So this is, I gotta watch out because that's gonna be the principle. This little diagram below. So this assumes that all traits are controlled by a single pair of alleles. This little comment, this example below, will be used later, but you can see here that these are all individuals. These are all different individuals here. So each individual in this image, each circle shows one individual plant, and the pair of alleles controlling its color. So we can see the green P here, that's controlled by these two green alleles. We can see the yellow P here, controlled by the yellow and green allele. We can see another yellow plant, controlled by an odd pair there. We can see the green plant and the green plant controlled by two green plants. So in the principle of genes in pairs, every single trait is controlled by one pair of genes. Only two genes are really controlling that trait. We're going to see much later that there are some examples where it's not a pair, but for the most part, for the majority of this course even, you can assume that a single pair of genes, a single pair of alleles controls this trait. Now, I do want to get straight here. I'm going to put a little sidebar here of the language. I think I have it come up a little later. A gene controls one trait. An allele is a form of that gene that gives a particular variation of the trait. So this implies, or this what I want you to be able to derive from this is that one gene may have multiple alleles, alleles, i.e. multiple variations. Yellow, green, color. Example. So a gene controls one trait. That trait may have multiple colors, multiple forms. Those forms are the alleles. So the allele is a form of that gene that gives a particular version of that trait. One gene may have multiple alleles, so which means multiple variations. There are some plants and organisms that have only really one common gene or one common form for an allele. The majority of human beings are carrying some sort of gene combination that gives out 5 fingers, and there are not very many there's not much variation in that gene in terms of developing 5 fingers. Now that could be a really complicated process. I don't know, but I'm just trying to give you an example. That would be kind of like a gene that everybody carries that has only one form. But there are lots of other genes that we carry multiple forms for. So hair color, we all have variations on hair, and the color of that hair, that natural color of that hair, and those would all be controlled by variations, very different alleles for that. Blood types, A, B, O, and AB. Those blood types are going to be controlled by many different alleles for the blood type gene. Okay, so if I mess up the language, that's why. It is a little tricky. Maybe I got a little bit of space down here. I'm going to just put that down here just so it's out of the way. I'm going to make sure this booklet gets posted up as well. So the principle of dominance and recessiveness is the concept that that all genes come in 2 forms. One form is the dominant form, and one form is the recessive form. And that recessive form is masked by a dominant allele. So the principle states that all genes have only two types of alleles. A recessive allele, and we can put in brackets form. A recessive allele and a dominant allele. We are later on going to talk about these complicated ones, so these are for the future. We can put these, that's a future conversation. So if we have, for example, so here's a chromosome, and when we're talking about alleles and genes are really just a spot on a chromosome. Remember that is the way we labeled chromosomes was very simple, by size. So chromosomes are defined by their size in the cell, and the genes are simply locations on those specific chromosomes. The alleles are then just the differences in the DNA at those specific locations, on those specific chromosome type. And that's what geneticists are doing nowadays, is they are looking at a chromosome. They might blow that chromosome up and look at all the different locations on it. They have ways of mapping those chromosomes, looking for specific regions that are common throughout different organisms and different animals, comparing those common features to between different organisms, different things, and then yeah, that's and then comparing those different sites and seeing if there's differences. So once they figure out that a specific location on a specific chromosome controls some trait, controls eye color or shape or size, then they go in and they try to see what kind of differences they find in those chromosomes between different organisms or between different individuals. And then they might even look at related species and see if they have similar chromosomes and similar alleles. Okay, so the alleles, so for an example, we could have the alleles capital T and little t. These are usually used for tall. The capital T will represent the dominant allele, which is uppercase, and then the lowercase t will represent the recessive allele, uh, which is representing lowercase, the recessive one, the one that is masked. So in this case, by the genes in pairs, so this is genes in pairs over here. I'm just going to type it over here and I'll move it. Genes in pairs tells us that for two different types of alleles, there are three possible combinations. Two dominant alleles, two recessive alleles, or one recessive and one dominant. That's it. It's a closed system. We can't have any other options. So this is why these principles are so key, because once you have these principles, once you have these assumptions, then you create a situation where there are limited possibilities. And that's what a good model is all about. So in science, a good model is one that puts limitations on, and then when the universe fits those limitations, then the model is good. When we find things that don't fit the model, then we have to start to expand the model and add caveats, add other assumptions or other language to the model to explain. So having anytime we have two of the same, we can label those as homozygous. So homozygous means both homozygous. This is the label that indicates that the two alleles are identical. So this could be TT or TT. I'm going to clean this up in a second. Excuse my, let's just change colors so it's a little bit more obvious. I'm going to move this over here. Shrink that. In the case of a heterozygous, hetero means different. So this is going to indicate, does not want to, let me type in there. Indicates that the two alleles are of different types. TT. And there's only one possibility. We do not consider order. So order doesn't matter in this case. So the order doesn't matter, but we do, through convention, always write the capital first. So that's a little convention there. So these heterozygotes, these are what we know as carriers, and homozygous are known as pure breeding. So these are pure breeding, or purebred, and these are the carriers. Remember, a lot of this language is, over 100 years old, maybe 200 years, almost 200 years old now since Mendel was born. Not 200 years since he made these conclusions, but so a lot of this language is old and brought forward from time of plant breeding and animal breeding. So we do use a lot of like pure breeding and carriers, languages from husbandry and other and farming. So carriers, these are heterozygous individuals. They carry a recessive allele, but because of our second principle of dominance and recessiveness, the dominant allele will mask the presence of the recessive one. So from our first principle, they're in pairs. They must be carrying two genes. But the dominance and recessive principle says that one of those alleles will overwrite or hide the other one. This implies that a carrier could pass a gene to their offspring without realizing that they were the carrier for it. So here is an unaffected carrier father, right, and an unaffected carrier mother carrying a recessive gene. Whatever let's label this red here. So here's our recessive gene, and they can both donate that recessive gene to their children, but in the rare case, they're both going to donate a recessive allele to their child, and the child now becomes a homozygous for this recessive condition. This homozygous recessive condition implies that they are now going to express the recessive trait. So this is the only child with the recessive trait. Only one with recessive trait. Even though they all carry it, this is the only one that can express it. So trait expressed. And so this is the difference between what we call the genotype and the phenotype of an individual. Because of the dominant recessive alleles, an organism's traits do not always reveal its genetic composition. Both parents in this case appeared unaffected by whatever this gene is, but they were both carriers for it. So their appearance, the expression of their traits, does not reflect their genetics, or doesn't fully reflect their genetics. So the phenotype. This is the expression, the overall expression of traits in an individual. How the individual appears externally due to their combination of alleles. The genotype is the specific combination of alleles found in the individual's genetics. So genotype has the word gene in it for genetics. Phenotype has the word pheno. Phrenology was kind of like looking at bumps on the head. Pheno is sort of surface and appearance in its origins as a word. So not sure exactly or getting deeper in that, but good enough anyways. So for example, if we had capital P here, this dominant P, and it represents the dominant allele for purple flowers, we could have a lowercase p that represents the recessive allele for the white flowers. As a side programming note, uh, the capitals or letter codes for alleles are always based on the dominant trait. So, and recessive letter codes are always the recessive, the lower case letter of the dominant allele code. So we don't, if they are recessive and dominant, then the dominant one will have a capital letter code based on what it does. So in this case, purple flowers cause purple flowers. The dominant trait causes purple, so we use a capital P. Even though the recessive trait causes white, the recessive trait is recessive to the purple, so we always label first by the dominant trait. Take whatever the dominant trait adjective is, take that first letter, and then lowercase letter that for the recessive trait, no matter what the recessive trait does. So in this case, if we had a genotype where we had two capital letters, we know that these are both purple-causing genes. These purple-causing alleles cause purple flowers to appear or to be expressed in the phenotype. The genotype, even if it was heterozygous, because it has one dominant gene, that dominant gene is enough to cause the expression of purple flowers. If no dominant genes are there, then we are talking about an expression of the white flowers, which is white. Now in the background, there's some good, we have a good idea, or we will have a good idea at the end about why this might be. Some ideas to think about is that when we have two copies of every chromosome in our cells actually really only need one copy to survive most of the time, or one copy to control the expression of traits. So these flower cells, on the flower, the cells of the flower, are accessing these chromosomes, and they really only need to access one of the two of them to find the instructions for how to make color in that flower. In the case of the white recessive gene, it's very likely that this is just a mutant of the purple flower, a flower gene, a mutant version of it that does not work and doesn't do anything. So when the plant is growing and it gets to the stage where it starts to request the formation of pigment, it sends enzymes to the DNA. They go and they start reading the DNA. In this first case, doesn't matter which chromosome they open up, they will find the information for purple. In the second case, the enzymes go to the DNA and they start looking for color. Half the time they find the purple color. Half the time they don't find a color gene. Well, it turns out in this plant that's enough. And a little bit of information on how to make purple is enough information to make the sufficient amount of pigment to make the plant colored purple. In the final example, the enzymes of this flower start to go and they start to read the DNA. They look for information and they can't find the information they need. They don't find information on how to make purple pigment. They might not even find any information at all. They go to the right spot on the chromosome and they find some convoluted, broken information, and that causes no pigment to form, which leaves the plant with a white appearance. Another option is that possibly they are finding the instructions for a pigment, and that pigment is white in color to the human eye, and that's also possible. All right. So that's one theoretical idea here on how this possible can go. And when we get to our DNA section, our last section of the course, you'll see how that could kind of play out. So we can now have some information on how to track this process, how these genes might work. We know from the earlier discussion that when we go to form the sperm and eggs, we only are able to donate one of two chromosomes. So, so in this donation, we are only going to be able to donate to our children, to our offspring, one of the two alleles that you carry. Genes in pairs says we have two alleles, and we are only going to be able to donate one of those two alleles to our offspring. So this creates a process where if you had two homozygous individuals with different colors, and you went and crossed them, that means that we would always be choosing one of these two for every single one of our offspring. That means that all of the children would receive one capital P and one lowercase p, or one dominant purple gene and one recessive white gene. And that would result in all of the flowers appearing purple. If we take all these purple flowers and we cross them, well, all of these flowers are heterozygotes. So that means that those flowers carry one dominant purple gene and one recessive white gene. And this is a little bit confusing, so I'm going to color this one in. So those are our purple genes there and our white recessive genes. So when they get crossed, this is what we call a cross here, or a Punnett square. I like these little diamond Punnett squares, honestly, but we'll usually do them on the ground. We can see that there is a possibility that you get both of the dominant genes. You could also get one dominant gene and one recessive gene. You could get one recessive gene from the other parent and one dominant gene from the other parent. Or you could receive both recessive genes from the same parent, or from not the same parent, from the different parents. And the result would be the children have one of these four genotypes. But the interesting thing is that two of these genotypes, or this genotype has two dominant alleles. This genotype has one dominant allele. This one has one dominant allele. So out of the four possible genotypes for this cross, only one of them has the white appearance, because only one of these genes in pairs is the homozygous recessive option. All right, so we're gonna just do a quick little tour of the last two principles. There is the law of independent assortment. So this is where the segregations of alleles for one gene occur independently of any other gene. So this is the idea that we are not dependent on, we cannot predict where the genes will go in the gametes. So here we can see an example that, one of the problems with this one is that they don't really so actually it looks like these pictures are backwards. I think this is the law of segregation and this is the law of independent assortment. There we go. That's why I was looking at this picture like, what's going on? This is another edit to be put into the pile here. Pictures of principles backwards. Okay. So the law of independent assortment is the idea of that alleles for one gene will independently assort, will occur independently to that of any other gene. So what this means is this is a good example here down below. Here we have our our different genes of all of our different letters. Or we have two different genes. We have a B gene and an S gene. And we can see that they are both there. This is someone who is heterozygous. So this one has a, the parent here has this type of genotype. They are heterozygous for B and they're heterozygous for S. And during the assortment, these genes get split up, and then these genes get shuffled and split up again. And at the end of this split up, we see that some of the gametes that they make have the two recessive alleles. Some of them have the two dominant alleles. Some of them have a recessive and a dominant, and some of them have a dominant and a recessive. And that all comes from this initial stage here, where we have an assortment through just like the random organization of these genes. We also have the crossover in prophase 1, which is not drawn here, but we would expect there should be some crossover here also that will randomize this as well. So this is enhanced by crossover. Let's call this independent. And then the law of segregations is that when gametes form alleles, when the gametes form, alleles are separated so that each gamete carries only one allele for each gene. I guess that's also kind of shown in both of these pictures. So neither of these pictures are particularly great. I think I can replace, maybe I'll just get rid of this one, and find a different picture. There is a better picture I'm trying to think of that's, yeah, that shows the independent assortment. But anyway, so the idea, we're assuming that every gamete has, not only does it have one of every gene, but it also has, those genes are randomly organized. So we are assuming assumptions indicate that every gamete is formed such that it only carries one copy of each gene, and the combination of alleles for these genes is unique for each gamete. So this is where, before we can, this is why identical twins are so rare. So this means that every sperm and egg is unique from its sister sperm and egg cells. So in meiosis, every single cell is different, and this was obvious to us from knowing now knowing meiosis, or it could be obvious to us now that we know our meiosis, but it is still an interesting concept that we can think about, as one that Mendel was able to put together from not knowing about meiosis and not being able to observe it. He would have to have made this assumption that when making the pollen or making ovums in the plant, each of those pollen are unique from each other, not only because they each carry one gene, but because they carry a unique combination of those genes. And so that's the conclusion of this lesson for today. Mendel discovers these traits through experiments with pea plants. He discovers dominance and recessiveness. The concept of homozygous and heterozygous individuals is now out there, as well as the concept of genotype and phenotype. I have never been one to just quiz you on the 4 principles so much, but they're not that hard to wrap your head around. They are important assumptions to understand when we want to go forward and discuss other examples. Good luck, and there is a video there about Mendel that will be showing up in the stream, that if you can watch that as well, that would be great. Later in the week, I'm hoping that we have one of the better meetups right now, or hangouts, so with a majority of our students in it. So I think we will try to host a little discussion in there, maybe later in the week, probably after we've done some practice questions, and then we can do the practice questions together, or after I've distributed some practice questions. So yeah, we'll talk about that tomorrow. Have a great day.